Alright kids, this is my first experiment with actually talking into a PowerPoint slide, but hopefully it'll go well for us. I'll try to be succinct. This first slide is my schematic of where the points are distributed for the test. You can see that there's kind of a heavy concentration on understanding what's haploid, what's diploid, in terms of the different stages of mitosis and meiosis. There's also all that nasty business about are they chromosomes or chromatids. You'll see that again. You'll have to identify the stages of mitosis in the onion root tip and the whitefish blastula slides. Similarly, with the ovary and testis tissues, you'll have to be able to describe the different cell types in that and relate what those cells what the different cells, where they are in terms of mitosis or meiosis. There is pretty much a buttload of genetics, so do all your practice exercises, learn about dominance, all the different types of those for sure, and I'll try to touch up a couple of those things here, but the problems are still up to you. And then finally with fermentation, that's where most of the discussion of the scientific method, interpreting things, is going to come in. Then there's also a couple of fine points, mostly about what the nature of the substrates are. Then what about glycolysis? So this is not exhaustive, but it's just to give you an idea. Alright kids, this is take 4. Yeah. So this slide is going to try to re-demonstrate the relationship between chromosomes, chromatids, haploid, and diploid during the major stages of mitosis and meiosis. Starting here on the right, in mitosis, you have a normal 2N cell, any kind of cell in the body, and its purpose is to self-renew itself. And so to do that, it has to undergo DNA synthesis, and that's what's represented by these asterisks and also the darker circles here and in meiosis. Once we have the DNA duplicated, it's still 2N, because what we're counting for N is the number of chromosomes, not the amount of DNA there. Okay, so briefly during anaphase, it's going to split up those sister chromosomes, those sister chromatids. They're going to become chromosomes themselves, and so very briefly it will be 4N, because all that duplicated DNA will now become individual chromosomes. But remember, this is the fine point on this, and really we're going to very quickly divide that back into two 2N cells. You can see down here. And so meiosis busts it up a little bit differently. Again, they start with a 2N cell, 2 copies of each set of chromosomes, and it's going to undergo DNA synthesis, and now we have 2 sister chromatids as part of each chromosome. But meiosis 1's job is to split up this homologous chromosomes, and when it does that, what we have, even though the DNA is still duplicated, is we only have 1 set of homologous chromosomes in each of the cells. So that's characteristic of a haploid cell. So it doesn't have an alternate allele for any of its genetic information. So meiosis 2 is more like mitosis then. Its job is to bust up the replicated DNA, bust up the sister chromatids. And when we do that, analogous to in mitosis, in anaphase 2, this 1N cell, which is duplicated, will very briefly split into 2 different chromosomes instead of 2 sister chromatids counted as 1 chromosome. So when those chromatids are split, it will very briefly be 2N. But then again, very quickly, through telophase and cytokinesis, we're going to generate 2 very earnestly haploid cells from that one. Okay, so I've tried to jerry-rig this figure on the left to demonstrate the sister chromatids so that each of the stages matches up. It still looks a little hokey to me, but again, your normal cell, and what we're representing here is that this cell only has 1 pair of homologous chromosomes. Therefore, it has a total of 2 chromosomes in its 2N state. After DNA synthesis, we'll have twice the genetic information. Everything will be duplicated, but it'll still be joined by these centromeres, and that's essential. So we have 4 sister chromatids, but only 2 chromosomes. Therefore, we are still 2N. Meiosis 1 is going to split up the homologous chromosomes, so now you can see, hopefully a little clearer, that there's only one set of information in each of these cells. So that's haploid, but it's haploid where the DNA has been replicated. Meiosis 2 busts those up, and you end up with something that we actually perceive of as haploid, so not replicated. So this slide just kind of puts a different face on that, and hopefully I'll remember to hit all the major points about the differences between mitosis and meiosis. So one of the big things is in meiosis, during prophase 1, the homologous chromosomes are going to match up, and that gives us our opportunity for crossing over. Okay, so from the same 2N cell and the same DNA replication, the molecules are roughly the same. So if we look here, we've got 3 duplicated blue chromosomes versus 3 duplicated blue chromosomes over here as well. But again, just them lining up is the major difference in prophase 1. Now, remember that unlike metaphase in mitosis, really should just be talking about meiosis 1 in total, but unlike in mitosis, the first meiosis is again splitting up our homologous chromosomes. So we've lined these guys up, they're not covalently linked in any way, and we're just going to pull them to other sides of the cell. And you can see these guys have already undergone the recombination via crossing over in prophase 1. In mitosis 2, that's when we actually split up the sister chromatids, and that's where you can see somebody like this red chromosome here. You get slightly different haploid cells, slightly different in terms of the DNA they contain. Like here, this has that little blue tip, which is different than which came from here, which is different than the allele that came from here. I want to stress that in anaphase 2, the cell does very briefly become 2N, where it was haploid to start meiosis 2. Okay, but it's only during anaphase and telophase where it has those sister chromatids busted up into fresh, new, and therefore double the amount of chromosomes it had before. But then cytokinesis is going to split that back up, and we're going to end up with our haploid cells down here. And I hope that I've broken that down sufficiently on this, so forgive me. This is try number 3 to get across the same point. But if we just follow the diploid versus haploid condition of the cell, that's this first line here, the number of cells that are present, the number of chromosomes, and I've used the example of human chromosomes, and the number of chromatids that are present down here at the bottom, and when those change. If we just trace through there, prophase 1, we've already undergone DNA synthesis, so we are 2N because we haven't undergone any cell divisions, but we are not 4N because we don't count the duplicated DNA so long as the sister chromatids are still attached. Okay, so what we have, 46 chromosomes, 92 chromatids, because they're still fused. Metaphase 1, we line up, not much changes. Anaphase 1, not much changes, but we're starting to separate the homologous chromosomes. Once we have completed cytokinesis, that's the way that I've calculated this down here, is after completion of cytokinesis, we now have 2 cells. We've separated the homologous chromosomes, so again, they're They're one end, they have only one copy of each piece of genetic material, but they are still duplicated. So you have two copies of the same, I guess. And so, because there are no homologous chromosomes in these two different daughter cells, you have 23 chromosomes, but it's still replicated. You have 46 chromatids. Now we begin meiosis 2. We remain replicated, but haploid until we get to anaphase 2. Anaphase 2, let's see, up here, well, going back to metaphase 2, everything's aligned and everything's attached. We're going to start pulling on this. Right now the sister chromatids are joined, but here in anaphase 2 we actually separate them. You have individual centromeres for each of the former sister chromatids. Because they're all split, we now have no chromatids, and because each of the paired ones before now has their own centromere, we now have 46 centrosomes, chromosomes, and we are very briefly 2N. However, again, telophase and cytokinesis are going to take care of that for us in a big hurry. Divide the cell into 4 cells total, each with only one copy of unreplicated DNA, each containing 23 chromosomes. Okay, so a substantial part of the test is still on identifying the cells and tissues in both the ovary and the testis. And so I just kind of wanted to walk through this one more time and remind you about the different stages of this. So this slide doesn't use exactly the same terminology that we used, but I think I can make it fairly clear. So the very primitive self-renewing cells, not really in humans because we complete all this before the woman's ever born. The earliest cells, the 2N cells, have that key suffix to them, gonium. In this case, the O denotes that it's going to be the egg. Those cells are the primitive. They don't use that term here. Primitive oocyte or primordial, which is surrounded by primordial follicle. And let's see, those cells are going to graduate once they start mitosis into the primary oocytes. And the primary oocyte is easily detectable by the primary follicle that surrounds it, and that only appears as a reorganization of the cells surrounding it into that kind of columnar epithelial cell layer. It's just a single cell layer, but you'll see kind of a thickening of the cells. As the oocyte progresses through meiosis, once we get into meiosis 2, we start seeing secondary follicle, and secondary follicle is characterized by both an increase in the number of follicle cells surrounding it, and as well as starting to see the appearance of some holes in that in those follicle cells. And so we don't really have anything else to go off of a morphology until all the holes are connected. That's when we're getting to a mature follicle and a real true secondary oocyte. This is also called the Graafian follicle. Okay, so there's kind of some really technical stuff that I don't really get about when it actually becomes an egg, but even during ovulation, once the oocyte is released, it's still considered an oocyte. I don't think it's actually considered an egg until it's fertilized, and you have a few more, kind of cell biology changes in there. So for our purposes, just considered a secondary oocyte from the time it's in the Graafian follicle. And of course, after ovulation, that same follicle tissue graduates to become the corpus luteum, which is responsible for releasing estrogen and several of the feedback endocrine communications that keep the pregnancy going. I want to point out to you that in this tissue there are very few cells that are actually haploid. It's really just the ones that have completed the second level of meiosis, and so all the supporting cells are obviously diploid, and then even most of the cells that are coming from the germ cells are diploid. So this is just a look at more advanced, they call it tertiary follicles, but we can call it a Graafian follicle. And so what you see is a nice big contiguous hole, just a few cell layers thick of follicles surrounding everything, and then the secondary oocytes in here, surrounded by the zona pellucida, which is this kind of red staining on the outside of the big germ cell. And these are all just different sections of it. So this one was sectioned very closely as if it was down right here into this plane. That's why the hole seems so small. But that's pretty typical of what you would see in a mature follicle and a mature secondary oocyte that's kind of through meiosis too. Alright, on to the male gonads. So I've kind of given you guys this crutch that these cells progress through meiosis as they go from the outside to the inside, and so they gain further maturity as they cross that boundary. So cross that path. So on the outside here is spermatogonium. Remember, gonium, that's the most immature. The spermato, in this case, it's because it's going to give us the sperm cells. And so these cells will actually be self-renewing, so they'll be undergoing mitosis. And that can be 1 or 2 cell layers thick. You'll see that there's a qualitative difference once you actually enter into meiosis. I'll show you on the next slide. But basically, if it's begun meiosis 1, then it's a primary spermatocyte. And so much as you know about meiosis 1, that tells you all you need to know about that cell. For all intents and purposes, we don't actually see the secondary spermatocytes, which are basically defined as being in meiosis 2. Because we don't see them because that second division happens so quickly. And so we get to haploid cells most of the way down the tissue here, and we start calling them different things. We've been calling them primary and secondary spermatocytes going down here, but we can call it a spermatid at this point too. So once we're haploid, we can be a spermatid, but a spermatid can have 2 different states of differentiation. That's a good word. So down here you can see like an enriched nucleus, and these can take 2 different forms. There can be like a little line, or there can be this kind of head. The head is more of a symbol of more maturity, and it's not actually a spermata. Dang it! I'm sorry. There's a term for the mature spermatozoa. It's not a mature spermatozoa. It's not a mature sperm until it actually has the flagellum, and then it can be released into the lumen here. So I think I should just move on and show you on the next one. So in this slide, a couple things you can point out quickly. So these are the extending flagella of very mature spermatozoa or spermatids. And you can kind of see there can be nuclei that are clearly spermatids, but they're not necessarily connected to flagella, so they're not necessarily at that level of maturity. So the secondary spermatocytes and probably the primary spermatocytes, we can just lump them together, and you can see that they have this qualitative look, basically more empty looking nucleus. These cells on the outside, it'd be hard to tell what's spermatogonium versus the very earliest stages of primary spermatocyte, but I think we'll just use this qualitative difference here where you see the darkest nuclei. and call those the spermatogonium. Those are the germ cells themselves, the ones that are 2N and the ones that are self-renewing through mitosis to eventually yield the primary spermatocytes and the cells that go through meiosis. You may be able to detect some prophase somewhere in here if you're really sharp, but again, just stick with the qualitative differences. That's going to be spermatogonium versus primary and secondary spermatocytes. Okay, on to genetics. So there's a couple things I wanted to make perfectly clear because I didn't really hit them in class that well. I think we know codominance or incomplete dominance. That's when we get a blending of the phenotypes, and so there's no problem with that. But I never really hit this phenotype here. So we can say codominance is any condition where both of the phenotypes are expressed. And if you look at this flower, the white is expressed, the red is expressed, there's no pink anywhere. And even though I can't really explain to you why one is expressed in kind of regional areas and the other is not, that relationship is codominant. Okay, so you can just you can kind of think of spots as that, as codominant. And the exception that we talked about the tortoiseshell cat because it's an interesting genetic phenomenon, but really that's something we should consider as an X-linked trait, not worry about it as a function of codominance. It does represent having two different alleles there, which is also relevant to codominance, but remember, the tortoiseshell cat, more of an X-linked thing than anything else. So one of the things, thanks to Kevin K for this slide, one of the things that I overlooked in lecture was this concept of epistasis, and that relates to any of these genes. We're talking about a multi-gene, a polygenic trait. So if there's 3 or 4 genes that are giving us a different phenotype, one of the things that can happen is you can get non-Mendelian ratios. So if you look at the actual phenotypes listed in this chart, 9 to 3 to 4, which doesn't make sense in terms of our normal genetics. If you have these different genes interacting, they can kind of step on each other's feet, and hence the name, and cause expression of different traits and modify. And this can kind of be an up or down phenotype. You can see traits that if they were left to their Mendelian selves, they would be gray, for example, but the other genes acting on this can tilt it to very dark gray versus very pale gray or something like that. And so how you would group them in terms of their phenotype could therefore, it could fall into different bins, and we'd have different Mendelian ratios. I'm not going to go over this slide. I'd encourage you to read it, though. Just the kind of questions we're going to be, the kind of responses we're going to be looking for once you're asked questions about, if you're given this data, what does it mean? Make a hypothesis, make a prediction. There is a difference. And then where you find independent and dependent variables on a graph and what they mean are still important. They'll remain important through the rest of the semester. Finally, there's one thing that I need to hit on, the fermentation lab. Yes, it is important that you keep track of the inputs and outputs of this. Glycolysis, we have glucose. It does have an energy requirement going in. It does have a net release of a net gain of, down here, a net gain of ATP. One of the outputs is the output is pyruvate. In addition to those high energy ATP molecules. And remember, NAD is involved in this becoming NADH. That's glycolysis. Fermentation, of course, takes 2 different forms that we discussed. One in the muscle keeps all the carbon atoms connected, but again, it requires NADH to put protons on this fairly oxygen-heavy compound here, pyruvate. And the other peels off a carbon dioxide, makes acetaldehyde, and then moves on to ethanol once we've, again, taken hydrogens from NADH and added them to the molecule. All that's great, but this concept of substrate level phosphorylation, what are we talking about? So the main goal of this is to give us our ATP outputs, and we know once we study respiration, that's what goes on in the mitochondria, what requires oxygen. Most of the energy that we get from that is actually by pushing protons around, as well as electrons, and letting them kind of fall downhill through this channel protein, and kind of turn it like you would turn like water going over a water mill would turn the mill and give you the work output of that. And so what happens is there's proton gradient in between the two membranes, and those protons flow in, and they turn that enzyme like a water mill, and that water mill kind of puts together ATP molecules, which can then be exported and used by the cell. So that's completely different than the ATP output that we're getting from glycolysis. The ATP output that we're getting from glycolysis is a function of jamming the glucose molecule and some ADP molecules and some phosphates into the enzymatic groove of the glycolytic enzyme, and then smashing them together, and using the energy from the lysis of the glucose to sew that phosphate and that ADP back together. Okay, so this is something that it's a function of all these things getting together in the groove of the enzyme, which is different than using the chemical gradient that the mitochondrion uses. And so that the phenomenon for here in the pocket is called substrate level phosphorylation. Finally, I hope we'll get a chance to do a lot of this during Saturday, but do all the genetics problems, look at what your peers did. I looked at the PowerPoint slides for the flashcards. A lot of them were getting wrong. I didn't proofread all of them. So see if you agree with your peers, see if you can find something analogous online, and just practice. Okay, and I suggest approaching it with some degree of basic problem-solving skills. So write out what you know first, try to find out what you're looking for, and then see how the unknown, see how to connect the dots. I can tell you one thing that I really want to cover when we do some stuff in lab on Saturday is make sure that you're listing the gametes across your Punnett square, and not random configurations of your two different alleles. But I think I'm going to leave it at that.